Hi guys, it's both of us again, and we're here to talk to you a little bit about biotechnology, which we talked about in class today. Remember that biotechnology is the direct modification of DNA of plants, animals, any living organisms really. And tonight we're just going to talk about one of the things that's used in some biotechnology techniques, and that's plasmid DNA. And plasmid DNA is found in bacteria. So we've talked about how bacteria has one long piece of DNA in the middle, like one big chromosome, but then they also have these other cool little circular DNA pieces floating around, and they actually pick them up from their environment, so that's pretty neat. And those plasmids then can be used to do different biotechnology techniques, and so we're going to do some of those in class, and there's some really neat applications. So we're just going to give you the basics here today.
Awesome. So the other technique that we're going to use is the technique of using a restriction enzyme. So we know enzymes are proteins. They come in and they can cut or splice things together. So the restriction enzymes that we're going to be using are going to come in and cut DNA at a specific sequence. So we're actually going to be using two next week. One is called BSTE2. And it's going to come in and it's going to find a length on the DNA that reads GGT and then N, so it could be any nucleotide or any base, and then ACC. And so it's going to cut that piece of DNA at the G. So remember, the complement of that sequence looks the same. So on our slide here, you can see that it will also cut on the G, and you can it will now have these little sticky ends that will hang over, and it will now cut the DNA at certain spaces. The other restriction enzyme that we're going to use is HIND3, and it finds the sequence AAGCTT, and it will also cut between the two A's, and again we'll have these little hanging sequences. We're going to practice this tomorrow using a plasmid DNA. You're going to be cutting that DNA into different sequences, and you're going to see that when you cut a circle, you're going to end up with some small straight pieces of DNA. And then after that, we're going to talk about how we're going to run those straight pieces of DNA on a gel or gel electrophoresis, so that we can be able to visualize or see what sizes those are and what the restriction enzyme actually has done. Because when you're cutting things in a tube, it's just a clear liquid, so you're not actually sure what's going to happen. So being able to see biotechnology in action. All right, have a great night.
See you tomorrow.
Bye.